Okay, thank you. I'll go straight to the point. First, thank you, the organizers, you, David, and Kim, and Jeff, and everybody that put together this this big meeting with 165 presentations. With my presentation, we are going to go back to remote sensing as we started this morning. And I would like to acknowledge first the collaborator and co-PIs on the NIFA proposal that these results are coming from, Dr. Mark Barrow, Rebecca Bennett, Kelly Chamberlain, Ning Wang, and Paxton, Peyton Paxton in particular. I plan on first starting with a review. So this is a big project that was related to development of advanced molecular and phenotypic methods for drought tolerance in peanut across 3 states and 2 environments, completely different. Dry environment in Texas and Oklahoma, and subhumid environment in Virginia. Today, I'm going to present the results only from Virginia. So we knew that identification of sources of resistance to biotic and abiotic stress is key for the development of improved cultivars. We also knew that direct phenotyping is slow. So my role and my team role was primarily related to the development of high throughput phenotyping methods from aerial imagery. And this is in summary what we have accomplished in an early study when we applied 2 3D models in ArcGIS to phenotype plant height with quite high accuracy. That paper has been published already. So the picture at the bottom of the screen, you can see how we collected plant height. Basically, it is from the entire row and the main stem plant height. And then the relationship between derived and manually measured plant height with the 2 models. One a little bit better than the other, but the first that has higher accuracy requires a little more computer work, whereas the other one is pretty much straightforward. And why is plant height important? Sayantan Sarkar, my former graduate student and now postdoc at the University of Tennessee, I'm very proud of him, reported in his thesis that plant height actually in our environment is important. Taller plants tend to wilt more, and also they have more tomato-spotted wilt. And the wilting I'm talking here is not necessarily because of drought stress. It is the experiment in this case was conducted in a dry, rain-fed environment. And in Virginia, we do receive quite a fair amount of precipitation. So I think that the wilting is just is rather induced by heat stress. Then, knowing that combination of remote sensing and data analytics can successfully substitute for the direct phenotyping, we used ordinary logistic regression to estimate peanut wilting. And we succeeded to do that. A paper just was published recently. And we did it with quite high accuracy, 90% from aerial imagery and 99% from proximal images. You can imagine from a drone, you lose some of the image clarity. And why is wilting important? Mark told about wilting in Texas not being as important as we found out to be here. Well, in our environment, Sayant, and again, this information on the screen is coming from his PhD thesis, shows that wilting does reduce photosynthesis, quantum yield of photosystem 2, and peanut pod yield. So in our environment, even the wilting due to high temperature effect and high humidity may affect photosynthesis and peanut yield. Later, we wanted to use machine learning algorithms, knowing that they can improve the accuracy over the stepwise regression. And we used in this particular paper, manuscript now in review, we use artificial neural network models to estimate leaf area index. Leaf area index is a measure of peanut vigor. And the data we obtained showed that indeed somehow machine learning can provide models that are a little more accurate than stepwise regression, for example. And I guess that the data that I plan on going into a little more detail, not extraordinary detail because I cannot, but a little more detail today is about determining the suitability of 48 vegetation indices to detect phenotypic differences for physiological and agronomic characteristics in peanut. On all these tests, we have used 103 accessions of the USA mini-core collection. Again, all these results were obtained in Suffolk, Virginia in 2017 and 2018 in a dry, rain-fed environment. The vegetation indices we used were collected with an octocopter. They were orthomosaic in PIX40 and analyzed in BreedPix and ArcGIS. And 24 were extracted from blue, green, red, and near infrared reflectance. 11 vegetation indices were developed from the color space indices, and 13 were from combination of the color space indices. We also wanted to look at the broad sense heritability. Okay, we have these indices to estimate physiology and agronomy of peanuts, but so what? Can they be useful for gaining a genetic progress? So we measured stem count at emergence, and then at about beginning flowering every other week until beginning seed development, and of course yield and sprouting on the vines in the end of the season. Now we measure plant height, lateral branching, NDVI, and canopy temperature depression on the ground. NDVI we related with the biomass production, and in the case and also the vigor of the plants. Canopy temperature depression is a well described in the literature, an indication of transpiration of the plants and therefore water use. We also recorded wilting on the ground, and pretty much at the same times with ground measurements, we collected aerial images. This is just how the plots are looking, 3 replications of 103 plots, and then how the software extracted the blue, green, red, and near infrared reflectance that helped us to calculate some of the indices. Before moving forward, I would like to show you this graph with the weather information to show you that the 2 years were quite different from each other and from the multi-annual. If the maximum temperature wasn't that much different, the minimum temperature was relative to the multi-annual temperature. And there were slight differences between 2017 and 2018 as well that made that the heat units accumulated for the first half of the growing season were higher in 2018. What happened? Way more development of biomass, okay? So therefore, even though the cumulative rainfall wasn't that much different between years for this part of the season, in between the rains, that huge amount of biomass needed more water and didn't have more, therefore suffered more. And in 2018, I'm going to show you, we had way more wilting on the plants. And then in 2018, in the later part of the season, we had a bunch of rainfall. What do you think that brought? Disease, a lot of disease. Therefore, yield was much lower in 2017, half of that in 2018 versus 2017. So no wonder why year effect was significant for all the measured agronomic and physiological traits. Genotype had also a significant effect, but not significant for sclerotinia, CBR, and thrips damage. And the interaction was not significant for wilting. The same varieties that wilted badly in 1 year or less were the same in the other year. Tomato spotted wilt virus, I would say sclerotinia blight, CBR, and thrips damage. Now, just to be able to present you so that you have an idea about the average across the varieties, I'm presenting this table here that is grouping the botanical types, and you can see that in terms of plant height, differences were more or less between the 2 years, but they were really big in terms of branching. Wilting was double, yield was half, and sprouting was also very, very bad in 2018 versus 2017. This table presents only the physiological agronomic traits that show that the botanical types were significant from each other. And for plant height, for example, vulgaris and fastigiata were the tallest and had the most branchability. Peruviana was the most wilted in both years and also had the least yield in both years, as opposed to hypogaea, for example. This shows botanical types were not statistically different for TSWV, southern stem rot, sclerotinia blight, CBR, thrips, but I'm still showing you the table to confirm that indeed disease was more terrible and the thrips was more terrible in 2018 than in 2017. Well, this is a heat map of the correlation of the aerially derived vegetation indices, and the vegetation indices are the rows, and the physiological and agronomic characteristics of the mini core in 2017. And I grouped those by 3 botanical types. I realized that for Peruviana, we only had 2 or 3 genotypes, so I excluded that for now. But the colors here show red color, negative relationships, green color, positive relationships, and the intensity of the color shows how the relationship was more intense. The association was higher, in some cases close to 0.9 coefficient of correlation. The yellow colors are showing not so much association. What it is important to observe is that actually in 2017, the associations were pretty much the same for the VIs across all botanical types. And they were more for stem count, plant height, and lateral growth, yield, and also for wilting and TSW. Less association we were able to find for the diseases, probably because we really didn't quite have a good differences among the genotypes, maybe. This shows the same, but for 2018, that very ugly year. And here you can notice that the intensities are less, so therefore the associations are less. They are not in the same direction for most of the traits. Still, some of them, in particular those at the bottom of the table, they were still high and in the same direction as 2017. So many indices to look at, only few to consider. In terms of heritability, this is for the agronomic physiological traits. Why I'm not surprised that yield has low heritability and in average. So I presented those by the stage of vegetation. You can see that some heritabilities and taking into account both years, data from both years, the heritability indices were significant, but were high, but they were only at certain vegetation stages, which means that it is important when the phenotyping is to be collected. In average, across all the vegetation stages, they are all low. So for plant height, for example, they are low up to full pod, when probably the maximum plant height was achieved, and etc. So I think that the message of this chart shows that it is important when to collect the when to phenotype, regardless whether you do it in an old way or in a new way using vegetation indices. However, if you're taking vegetation indices across different stages and you use the average of all vegetation stages, based on the heritability data that I have here, it looks like you are probably more successful to get the differences that will increase the genetic gain that you're looking for. Also, good stages to take vegetation indices as a phenotyping tool looks to be for the most vegetation indices beginning a PEG and pod development. Anyways, in conclusions, significant variation for the phenotypic, agronomic, and aerially derived vegetation indices among genotypes and botanical types of the mini cores was identified in the subhumid environment here in Virginia. Aerial VIs were associated with the physiological and agronomic characteristics with different intensities depending on the year. So not every year is so good for phenotyping. I mean, phenotyping may be less successful like in a year like 2017 in comparison with 2018 in comparison with 2017. And some traits can be estimated with more success than others. For example, crop stand, height, and branching, and even yield were much better estimated by vegetation indices than disease, for example, and sprouting. Broad sense heritability varied with the trait and the growth stage when was collected, but certain VIs were significantly correlated with the physiologic and agronomic traits in both years. And I'm providing here two examples and their formula. They have been published, and they are normalized difference, CIE lab index and CIE lab index. A's, B's, and UNVs are nothing else but how the image, like you're looking at the screen of TV, how that a general color of that image changes A and U from green to red, and B and V to blue and yellow. So that's pretty much what they indicate. And that slight color seemed to be consistent in both years. They also had high heritability across the board. So therefore, probably aerial sense, physiologic and agronomic characteristics for breeding and precision ag applications could be successful. I would like to close my talk thanking my co-workers, Sarkar and Kazanov, NIFA collaborators, and the sponsors, Virginia Crop Improvement and NIFA Africa.
Yeah, thanks, excuse me, Maria, for a great presentation. There'll be some questions coming. So we're going to keep moving along due to time.